Biology, this video is an overview of photosynthesis. The goal of photosynthesis is to convert solar energy, the energy found in sunlight, into chemical energy, the energy found in the chemical bonds of organic molecules like sugar. On the bottom of the screen here we see the equation for photosynthesis. Photosynthesis is an endergonic process. It's an anabolic pathway. So you need an investment of energy, and the energy that powers photosynthesis is going to be sunlight. The reactants are water and carbon dioxide, and the products formed are going to be sugar. Here you see glucose, and oxygen is going to be released as a waste product. Where does photosynthesis take place? Photosynthesis takes place in a number of different organisms. Plants photosynthesize. You have green algae that photosynthesizes. Certain bacteria like cyanobacteria photosynthesize. Here we concentrate on a plant. The photosynthetic organ of a plant is the leaf, and the leaf is going to contain cells called mesophyll cells, and these mesophyll cells perform photosynthesis because they contain chloroplasts. In order to allow gases to enter or leave the leaf has holes in the bottom of the leaves mostly called stomata. And each stomata is going to be flanked by a pair of guard cells. You see them right here and right here. And these guard cells control whether or not the stomata are open. The stomata is going to be how carbon dioxide gets in for photosynthesis and how oxygen leaves. Besides carbon dioxide and oxygen entering and leaving through the stomata, water also has the ability to evaporate out of the stomata. The evaporation of water out of the stomata is called transpiration. Remember, the evaporation of water out of the stomata creates transpirational pull, the force that pulls water up the xylem of a plant to get to the leaves that need it for the process of photosynthesis. If you look at this picture of a chloroplast, inside the chloroplast we have these pancake-shaped discs. We call them thylakoids. A stack of thylakoids makes up a granum. A particular chemical reaction is going to take place within the thylakoids. The fluid outside of the thylakoids is called the stroma, and a different chemical reaction is going to take place within the stroma. Here's an overview of the two chemical reactions that take place within photosynthesis. Chemical reaction number one is the light reactions. The light reactions take place within the thylakoids. Reaction 2 is the Calvin cycle, and the Calvin cycle is going to take place within the stroma. The two reactants of the light reactions are going to be light and water. During the light reactions, water is going to get oxidized. So it's going to lose its electrons, it's going to lose its protons. When it loses its electrons and protons, it's going to cause oxygen gas to be released as a waste product. The electrons that came from water are going to be transferred to NADP+, and is going to form NADPH. NADPH is an electron carrier or an electron shuttle that's used during photosynthesis. As the electrons move, the movement of electrons is going to be used to generate ATP, another product of the light reactions. The ATP and the NADPH made during the light reactions are going to leave the thylakoids and enter the stroma, and they're going to be the fuel source that powers the Calvin cycle. The major reactant in the Calvin cycle is going to be carbon dioxide. During the Calvin cycle, carbon dioxide is going to get reduced. It's going to be given electrons, it's going to be given protons. The electron donor is going to be NADPH. So during the Calvin cycle, NADPH is going to get oxidized. It's going to lose its electrons, and those electrons are going to be transferred to carbon dioxide. When carbon dioxide gets reduced, the end product is going to be sugar. It's going to be glucose. The ATP is used to fuel the Calvin cycle, to power all of its activities. Here we see our major end products. 1, oxygen, which is really just a waste product, and 2, sugar. This is the chemical energy that has been created through the process of photosynthesis. To understand photosynthesis, we first have to understand sunlight. The sun emits radiation in different wavelengths. Different wavelengths of light correspond to different colors. The color of an object is not determined by what wavelengths are absorbed, but instead by what wavelengths of light are being reflected. So the thylakoids are green, and that's because they contain a pigment called chlorophyll. Chlorophyll is going to absorb red light, it's going to absorb blue light, it's going to absorb purple light. The wavelength of light that chlorophyll reflects is going to be green light. Right? That's why we see plants, and that's why we see chlorophyll as being green. Here you see the absorption spectrum of a chloroplast. The chloroplast is going to contain numerous photosynthetic pigments. Pigments are molecules that absorb very specific wavelengths of light. This diagram shows the 3 major pigments. You have chlorophyll A, chlorophyll B, and you have carotenoids. And it shows you the wavelengths that they absorb and the wavelengths that they reflect. You can see that if we put all 3 of these pigments together, generally speaking, they absorb purple light, they absorb blue light, they absorb red light, they absorb some orange light, and the light that they don't absorb at all is green light. Green light doesn't get absorbed, instead it gets reflected, which is why we see a plant as being green. What does this all mean? This means that photosynthesis would occur less under green light. Photosynthesis wouldn't really occur at all under green light because plants can't absorb green light. They can't do anything with that energy. Photosynthesis occurs the best under blue light, purple light, or red light. Here we look at the individual reactions of the light reactions, and this exact picture here shows us non-cyclic electron flow, and we'll understand what that means in a minute. This picture shows one specific thylakoid. Here you see the thylakoid membrane. The space inside the thylakoid is called the thylakoid space, and outside the thylakoid we have the stroma. The light reactions are going to utilize 2 photosystems. You have photosystem 2, you have photosystem 1. Each photosystem is made up of a light harvesting complex, that's what we see in darker purple right here, and a reaction center. Attached to each light harvesting complex is going to be an electron transport chain. You see one right here, and you see one right here. The light reactions begin with photosystem 2. The light harvesting complex is going to be made up of multiple chlorophyll pigments. You see them all here in green. And all those chlorophyll pigments absorb wavelengths of light, and they funnel all that sunlight energy to the reaction center. At the reaction center, water gets oxidized. It's going to lose its electrons, it's going to lose its protons. When it loses its electrons and protons, oxygen gas is going to be released as a waste product. The electrons that came from water are transferred to the reaction center. The reaction center is made up of a pair of very special chlorophyll A molecules. When all that solar energy gets funneled to the reaction center, it causes the electrons that came from water to jump up to a higher energy level. And when they jump up to this high energy level, they get grabbed onto by a protein called the primary electron acceptor. That protein is the first protein in the electron transport chain. These electrons that were energized by light then move down the electron transport chain, and just like in cell respiration, the electron transport chain contains proton pumps, and these proton pumps pump all the protons that came from water into the center of the thylakoid. And what we're building up here is a hydrogen ion gradient, also called a proton motive force, just like putting water behind a dam. When all these protons build up, they're going to rush through an enzyme in the thylakoid membrane called ATP synthase. When the protons rush through ATP synthase, it's going to spin a turbine, and the spinning of the turbine is going to be used to regenerate ATP. So photosystem 2 is used for the production of ATP during the light reactions. When the electrons get to the end of the electron transport chain of photosystem 2, they're really low in energy, and they go to the reaction center of photosystem 1. Well, the light harvesting complex is going to absorb more solar energy, and it's going to funnel all that solar energy to the reaction center, and it's going to re-excite those electrons. Again, they jump up to a higher energy state, get grabbed onto it by a primary acceptor, go down a second electron transport chain. Waiting at the end of this second electron transport chain is going to be NADP+. NADP+ gains the electron. It also gains the proton that went through ATP synthase, and it becomes NADPH. NADPH is made specifically during photosystem 1. We call this whole process non-cyclic electron flow because the electrons flow in a direct line. They begin with water and they end with NADPH. Non-cyclic electron flow makes equal amounts of ATP and NADPH. We have a problem though. The Calvin cycle, which we're going to get to in a minute, needs more ATP than NADPH. In order to just make the ATP, the light reactions are going to convert from non-cyclic electron flow to cyclic electron flow. Cyclic electron flow is going to turn off various parts of the light reactions, and it creates this loop where electrons are continuously energized by photosystem 1 and go down the electron transport chain of photosystem 2. Well, we know the electron transport chain of photosystem 2 is responsible for the creation of ATP. So the thylakoids are able to convert back and forth from non-cyclic electron flow to cyclic electron flow in order to control the production of ATP and NADPH. The ATP and NADPH made are going to travel into the stroma, which is where the Calvin cycle takes place. The major reactant of the Calvin cycle is going to be carbon dioxide. Carbon dioxide gets fixed, gets grabbed onto by an enzyme called rubisco. When carbon dioxide gets fixed by rubisco, it gets combined with another molecule called RUBP, ribulose biphosphate, and they make this product, which is not really important for the overall understanding of the Calvin cycle. Phase 1 is going to be carbon fixation, the process by which rubisco takes in carbon dioxide and combines it with RUBP. Phase 2 is going to be reduction. This is going to be when NADPH gets oxidized, loses its electrons, and gives its electrons to this molecule right here called PGA. So the important part of this second phase is that carbon dioxide is getting reduced. It's being given electrons. The end product is going to be this 3-carbon sugar called G3P. 2 G3Ps can be used to make 1 glucose. After the end product has been made, the last phase is going to be the regeneration of RUBP. The Calvin cycle is called a cycle because this RUBP is constantly being recycled over and over again. Something to note here is that the Calvin cycle is going to utilize 6 NADPH and it's going to utilize 9 ATP. This is going to be why the light reactions have to convert from non-cyclic electron flow to cyclic electron flow in order to make the extra ATP that's needed to regenerate RUBP. So just to look at this Calvin cycle, right, carbon dioxide is going to enter, and it's going to be grabbed onto by an enzyme called Rubisco. When carbon dioxide enters the Calvin cycle, it gets reduced by NADPH. NADPH is giving carbon dioxide its electrons, and it's giving carbon dioxide its protons. The end result is a 3-carbon sugar called G3P. Every 2 G3Ps is going to be used to make 1 glucose. Finally, the starting material, a molecule called RUBP, is going to be regenerated at the end of the Calvin cycle. Here we see our major product of photosynthesis, glucose. Glucose contains chemical energy stored in its chemical bonds. Normal plants are called C3 plants, and C3 plants encounter problems on hot, dry, and windy days. Heat, dryness, and wind all increase the rate of transpiration. Remember, transpiration is the evaporation of water out of the stomata. If too much transpiration takes place, a plant can get dehydrated and it could die. So to prevent excessive transpiration, the stomata close. There's a problem though when the stomata close. Carbon dioxide can't get in for the Calvin cycle and oxygen gets trapped. Well, there's a problem. Rubisco has the ability to also fix oxygen as well as carbon dioxide. But the problem with oxygen is it doesn't contain any carbon. Without carbon dioxide, you cannot produce glucose. So if the stomata close and oxygen begins entering the Calvin cycle instead of carbon dioxide, a really wasteful process takes place called photorespiration. Well, how then do plants live in hot, dry, or windy conditions? Well, there are 2 types of plants that have special adaptations that prevent photorespiration from taking place. They're C4 plants and they're CAM plants. C4 plants typically live in the Midwest. Here you see an example of a C4 plant. You have things like corn is a C4 plant, sugarcane is a C4 plant, and the other type of plant is a CAM plant. CAM plants tend to really be the desert plants. Both C4 plants and CAM plants do one thing in common. They have the ability to store carbon dioxide. If you can store carbon dioxide, you do not have to worry about photorespiration taking place. Because no matter what, whether your stomata are open or closed, carbon dioxide has the ability to enter the Calvin cycle. The difference between a C4 plant and a CAM plant is how they store the carbon dioxide. C4 plants store it as a 4-carbon molecule. CAM plants store it as an organic acid. In addition, Cam plants, which tend to be more extreme and live in the desert, they only open their stomata at night when it's much cooler. The stomata are always closed during the day. That completes this video.